All right, since we missed class today due to the assembly, you guys had requested a video of the notes. So we'll do a quick video. Please do complete your notes outline that is on Canvas on genetic mutations, specifically chromosome mutations, and then we'll briefly go over the basics of the mutations in class tomorrow. So mutations are a change in the genetic material that is permanent. So if there's a change in the material in a cell and the cell divides and passes that information down to a new cell, whether it's a cell that's made by mitosis or meiosis, that new cell will then have that change. It will remain in that cell, and then new cells that are made. So these changes can occur in our somatic cells. If they're in our somatic cells, remember our cells divide by mitosis, and they would pass down to the new cells, but remain within that organism. If these cells are dividing by meiosis and making our gametes, then that mutation would be passed on to the offspring if those gametes are used in fertilization. So that new offspring, the new organism, will then have a mutation in all of its cells. So chromosome mutations impact either the entire chromosome or a piece of the chromosome. So it's going to be a change in either the structure of that chromosome or how many chromosomes we have, so an extra chromosome or missing, as we saw in our karyotype. So this might involve the loss or gain of an entire chromosome, or it might involve the loss or gain of a piece of chromosome, or like we said, a change in the structure. So we might see a piece of a chromosome be relocated or be lost. So here are some mutations. So our chromosome mutations come in a couple of forms. There's a chart for you to fill out, but let's just look at the names at first. Deletion, inversion, duplication, translocation, and insertion. Each of these we'll discuss in turn. Notice that for deletion, inversion, duplication, these terms are familiar. So take advantage of the opportunity that the terms are these familiar terms. So you can imagine that deletion means we'll delete a piece of a chromosome. That is not always the case with science terms, so take advantage of it. Let's talk about each. So in the chart, you're writing in the name of the mutation. So in this case, it'll be deletion on the left, and on the right-hand side, the description, which usually are pretty brief. So in a deletion, we're going to see that a piece of a chromosome breaks off, and that piece is lost. So when we say that piece is lost, it will no longer be found in that cell. If a cell divides, it might end up in the other cell, for example, but it's not maintained where it belongs attached to that chromosome. So notice in this picture here, so our chromosome is purple, and we're highlighting this pink piece just to track it. So that piece might break off. Remember, chromosomes are flimsy and flexible structures, so they could get tangled and break. And that piece is broken off, the rest of the chromosome reattaches, so now we have a piece missing, and therefore that is deletion. Our second chromosome mutation is called inversion. So to invert means to flip upside down. So we have a piece of a chromosome that breaks off, but then that segment inverts or flips and reattaches. Best to look at a picture here. So again, we have that purple chromosome, and the color code is just to help us track it. So we have a pink piece and a green piece. That piece breaks off and then flips, maybe it gets tangled with something, flips around, so inverts, and then reattaches. Notice the greens at the top and the pinks at the bottom. So what's the problem with this? It still has that piece of chromosome. But this is very confusing for the cell. Just like you read left to right, the DNA is only read in one direction. So if this information is flipped, now the information is on the wrong side and backwards. So it'd be less like us taking a piece of what you're reading and half of that paragraph is flipped upside down and you still need to read it left to right. It won't make sense. It'll just be gibberish. Duplication is when we have a duplicated piece. So a piece or gene segment or sequence is repeated. So say DNA replication occurred and we copied this segment twice, we would have an extra piece of that chromosome. That can be very confusing. Translocation is when we have a piece of a chromosome break off of one chromosome and then reattaches to a different chromosome. So trans means to move and location is where it belongs, right? So let's say this is chromosome 1, this is chromosome 20. Piece breaks off of chromosome 1 where that's lost, and now it is attached to chromosome 20. That is called translocation. Now this is similar to what we saw for crossing over. However, in the case of translocation, we're most likely looking at non-homologous chromosomes, so chromosome 1 and chromosome 20. In crossing over, we're looking at a fair trade between homologous chromosomes. Keep in mind, this could happen as an unfair trade in homologous chromosomes as well. All right, finally, insertion. Insertion will look a little bit like translocation because we do involve two different chromosomes. In this case, the piece is broken off of one chromosome and attached to another, but it's inserted. So the term insertion, that piece breaks off. It could be a middle, could be an end. This is chromosome 4, and this is chromosome 20. So a piece breaks off chromosome 4, and in this case is inserted in the middle of chromosome 20. This could also be due to crossing over that is unequal. So if we did have homologous chromosomes, so chromosome 4 and chromosome 4, and they traded a piece, but it wasn't a fair trade, it was a smaller piece from one chromosome and a larger piece from the other, that would also lead to insertion. Now, each of these leads to a disorder, but the disorder that it leads to will vary depending on which chromosome we are talking about, which genes we are talking about, how much of that chromosome has been mutated. So the disorders it will lead to will vary greatly depending on which chromosome pieces we are talking about. So a quick animation. Here's the original chromosome, and we'll walk through each of them. So inversion is if the chromosome is flipped. Notice a piece broke off and flipped. Translocation, a piece broke off and attached to another different chromosome. Duplication, we have an extra piece added. Here's our original again. Deletion, we lost a piece. And this just rolls through a couple of times. Notice the flip for inversion. Translocation, 2 different chromosomes. The only one that's missing in this animation is insertion, where a piece breaks off of one chromosome and inserts in the middle of another chromosome. So you have a practice on the second page where you are given this original chromosome, and you are asked to try to identify what mutations are happening in relation to this. So you look at the second, third, and fourth pictures and compare them back to the original to see if you can identify the type of mutation. So I would ask that you pause the video now to see if you can identify those and then restart the video. It will only take you maybe 15 or 20 or 30 seconds. So let's look at the first one. So notice that A, B, C, D, E, F, G, that represents either genes or gene segments. It could be a cluster of genes. Keep in mind there are many genes on any given chromosome. Notice that D was added twice, so we have a duplication, and E is missing, so we actually have a deletion as well. In the second one, notice we are missing that segment labeled D, therefore this is deletion. And then finally, D, E, and F were broken off and flipped, therefore this is inversion. So that shows you how a chromosome can change structure. Now, when we looked at our karyotypes, we had either an extra chromosome or a missing chromosome as a possible genetic mutation, and that would be a chromosomal abnormality. And we saw that in our karyotypes, where we had the extra of chromosome number 21. Let's take a look at how that happens. Now, we did mention this, and it's called non-disjunction. Non-disjunction is kind of a strange term. So let's take this apart a little bit. So a junction is where things meet. To disjunct means to come apart. So our chromosomes should come apart. In anaphase 1, we have homologous chromosomes separate. In anaphase 2 or mitosis, we have chromatids separate. So this should happen. In the case of non-disjunction, they fail to separate. So non-disjunction is the failure of chromosomes to separate during meiosis. Specifically anaphase 1, but technically it could be during anaphase 2. Really could even happen during mitosis in anaphase. However, we're looking at passing this off to our offspring. So when chromosomes fail to separate, remember, the two chromosomes are supposed to separate, one X to one side, one X to the other. But if they fail to separate, both will go to one side and neither will go to the other. If either of those is used in fertilization, now we have a new cell that either has an extra chromosome, 47 instead of 46, or a missing chromosome, 45 instead of 46. Okay? So that's going to cause our gamete to have too many chromosomes or too few chromosomes. Again, that's only if the cell is used in fertilization. Let's see what this looks like. So this is a cell in metaphase 1 of meiosis, where homologous chromosomes are paired and lined up along the middle. So this is normal. In our next cell, we see anaphase 1, and our homologous chromosomes are separating on the left as they should. On the right, they're failing to separate. So notice both chromosomes are going to one side and neither is going to the other. This is non-disjunction. Notice in our cells, so this is anaphase 2, where the chromatids are separating, we end up with way too many chromosomes. So we should have 4 chromosomes going to 2. Remember, in meiosis, we divide twice and end up with half the number of chromosomes. But instead of having 2 chromosomes, we have 3 in 2 of our cells and only 1 in the other 2 cells. If any of these are used in fertilization, we're going to end up with a cell with too many or too few chromosomes after fertilization. Let's look at some examples. Down syndrome is the example that we saw in our karyotype. We have an extra of chromosome 21. It's called Down syndrome or trisomy 21. So trisomy means 3 of 21. This leads to a disorder that you are familiar with most likely. It's called Down syndrome. It's relatively prevalent in our population. Notice their 2N number is 47 versus 46. A couple of other examples. Notice that the location of the mutation does offer a different disorder. So Klinefelter's, notice we have, instead of having one X and one Y or two X's, we have two X's and a Y. So we have an extra sex chromosome. This individual has what's called Klinefelter syndrome. Again, their 2N number is 47 instead of 46. Klinefelter's is a disorder where the individuals have both male and female reproductive structures. We would consider this person most likely a male because they do have the Y chromosome. But those reproductive structures are underdeveloped and do not allow them to produce offspring, so they are infertile. Another example that also happens at these sex chromosomes is called Turner syndrome. Notice mono means one, so monosomy. So notice the rest of the chromosomes are aligned as we would expect. However, our final chromosome set here should be two sex chromosomes, either XX or XY, and we only have a single X. Again, this individual has 45 chromosomes as their 2N number because they're missing a chromosome. This individual would be female because there is no Y to indicate a male. Okay, so this individual is female, but they have what's called Turner syndrome. Turner syndrome leads to an individual that has shorter stature, they're infertile because they have underdeveloped female reproductive structures, and they have a few other characteristics as well. So these are just some examples of how non-disjunction can lead to a specific disorder due to the extra or missing chromosome. You have also a very brief constructor response question set to do, so please make sure you try to answer those questions. Again, the answers can be short. We will go over those tomorrow. We'll do a quick overview of chromosome mutations tomorrow, and then a quick overview of the constructor response as well.